What causes someone to have too many or too few chromosomes? We know from our study of mitosis in the previous unit that if we had a cell that contained one DNA molecule, that when that cell makes a copy of its DNA, and those two DNA molecules locate one another to form the duplicated chromosome, that the goal of mitosis is to transmit one of those DNA molecules into each of two new cells. That way, each of the new cells will have the same amount of DNA as the original cell. This is what should happen during mitosis. However, if we think about that same cell with its one DNA molecule undergoing DNA replication, and those molecules finding one another to form the duplicated chromosome, what would happen if these chromatids didn't separate? Well, it's entirely possible that when that cell divides, that one cell would inherit 2 copies of the DNA molecule, while the other cell would inherit 0 copies of the DNA molecule. It's not normal for a cell to inherit an abnormal amount of DNA, whether it's an extra DNA molecule or if the cell were to inherit no DNA. And we saw in the previous unit that this is one of the reasons why normal cells can become cancerous, because they inherit an abnormal number of proto-oncogenes or tumor suppressors from the original parent cell, thus making it more likely for that cell to transform into a cancerous cell over the course of time. But what could cause something like this? At this point, you are familiar with the concept of a karyotype, or a chromosomal spread that shows the chromosomal distribution within a typical cell. This is the chromosomal spread, or a karyotype, from a human cell. We know that it's a human cell because there are 23 pairs of chromosomes. Here's the 22nd pair here, plus the XY pair. We know this individual is female because of the 2 X chromosomes, but we can see pretty clearly that this individual has an abnormal number of chromosomes 8, an abnormal number of chromosomes 20, as well as an abnormal number of chromosomes 12 and 17. The rest of the chromosomes all appear in pairs, as you would expect in a normal karyotype. So what caused an individual to have this number of abnormal chromosomes? It's unlikely that this individual could have inherited all of these chromosomes and their abnormalities as a result of mitosis. Instead, it's more likely that this individual started out with a chromosomal abnormality as a result of meiosis. Let's look at that next. In order to fully understand how an individual could inherit that abnormality of chromosomes, we really have to focus on our understanding of meiosis and the production of sperm and egg. You know that it's possible, just like in mitosis, for chromosomes to fail to separate during meiosis as a consequence of a non-disjunction event. So it's possible that pairs of homologous chromosomes may not separate during anaphase 1, consequently producing cells with either an extra chromosome or cells that are missing a chromosome altogether. But it's also possible for chromosomes, or in this case chromatids, to fail to separate during anaphase 2 and produce cells with an abnormal number of chromosomes. In this case, or in this example, 2 of the 4 cells typically produced during meiosis would have the correct number of chromosomes. Notice we have 4 chromosomes here, and we have 2 chromosomes there. And we know that meiosis always produces cells with half the number of chromosomes of the original cell. So how can we explain, using our knowledge of meiosis and our understanding of non-disjunction, how an individual could inherit an abnormal number of chromosomes, thus setting the stage for all of the cells in that organism to have an abnormal number of chromosomes? Maybe not quite as severe as the image that you saw previously, but something along those lines. Well, if we think about an individual on the left-hand side of the screen, let's say this is the female, and on the right-hand side of the screen we have the male. Well, let's say we want to produce an individual that has 3 chromosomes. So this parent and this parent are each going to produce egg and sperm respectively, with an abnormal number of chromosomes that would produce a fertilized egg at conception that contains an abnormal number of chromosomes. Because that fertilized egg will then divide through mitosis, all of the cells that are the result of mitosis we would expect would have the same number of chromosomes as that original fertilized egg. But how do we get to that fertilized egg with the abnormal number of chromosomes? Let's see if we can figure it out. Well, let's keep the numbers simple and assume that in this female we have 2 chromosomes as well as in this male. Well, if we wanted to get to a fertilized egg with 3 chromosomes, it should be pretty easy to do. We would allow for a normal meiotic event so that at the end of meiosis 1, each of the 2 new cells has 1 duplicated chromosome. And then at the end of meiosis 2, each of the new cells that are produced would have 1 chromosome, or 1 DNA molecule. That takes care of the male. But what about the female? Well, if our goal was to get to 3, there's different ways that we can get there. Let's look at one of those possible ways. Let's say that meiosis 1 occurs normally, and each of the 2 new cells produced at the end of meiosis 1 have the 1 DNA molecule. But, as a consequence of a non-disjunction event during meiosis 2, one of the cells that's produced has 2 DNA molecules, while the other one has 0 DNA molecules. And for the purpose of simplicity, let's assume that this cell on the right-hand side inherits the correct number of DNA molecules. Well, we can see pretty clearly that what we've produced in the female is an egg cell with 2 chromosomes. And in the male, we've produced a sperm cell with 1 chromosome. If, at fertilization, these 2 cells were to fuse together to produce the fertilized egg, then the fertilized egg would contain 3 chromosomes. And thus, as their mitosis took over, all of the cells that are the progeny of this original fertilized egg would also have 3 chromosomes, thus setting the stage for an organism that has a complete collection of cells, a complete population of cells with 1 extra chromosome. So now you try it. Can you come up with a way to produce a fertilized egg with 3 chromosomes that's different from the way that I've described here on the screen? If you can, then you're well on your way to solving the karyotype practice problems that have been posted to the class website.